for being here, and thank you for attending this session on the topic 2 of the IMI 2 call 1 on retinal disease. So the purpose of this workshop really this afternoon is for you to get more acquainted with the topic text, but especially to give you the opportunity to ask any questions, and we greatly to have the topic writer, the coordinator from the industry side. So that will be your opportunity for really ask the questions to help you to formalize your application. And this workshop is really done in a way that it's transparent for everyone, so everyone will have the same information to help you to really finalize or to apply for this topic. So I may wait a little bit until everyone enters. Okay. Now we have 2 hours in this session, which is long, but at the same time I'm sure it will go fast. So the way we will split the session is as follows. We will have first the presentation from Oliver Zeitz, who is the Vice President and Head of Global Development Ophthalmology at Bayer Healthcare. And prior to joining Bayer in 2009, Oliver was a senior physician in ophthalmology at the University Hospital of Hamburg, as well as assistant professor at the Medical Faculty of Hamburg. So I think having both the hat from industry, but as well having the academic background and the clinical background will really put certainly Oliver in a great position to explain on the need of this collaborative approach to tackle this research area. After the presentation, we'll have plenty of time for question and answer. So please, this really will be the time for you. We'll have probably 1 hour to go through any question you may have. And we will see as well the question coming from the people who are looking from the web stream, whether we can address all the questions. We will close the session by a 30 minutes elevated pitches. So when you register, you were asked whether you wanted to present in a 90-second a statement about your organization and what it could bring to an applicant consortium. We have received 11 requests, and therefore we will go one by one to these elevated pitches. So as I said, it's a very short and quick presentation and summary of what the organization can do. If anyone wants to do it, even without having said so at the registration, we'll try to find a way to accommodate, but please keep in mind that this was asked at the time of the registration. Now, before we go to the presentation of Oliver, I just want to remind you of something which is important and that was already alluded this morning several times, is that the call is still under the deadline for submission of the application is not yet passed. It's in November. So there's still time for every one of you to apply to join a consortium. So there is really this feeling that, as was mentioned this morning, there is no pre-cooked. It's not yet done. It's a competitive call. Everyone has this chance. By the deadline, after the deadline for the submission, obviously, we cannot accept more application. But again, the application will be reviewed by independent expert. And therefore, as we already had in IMI 1, everyone has its chance if the consortium has addressed all the objectives of the call and as well has read all the document in order to qualify for an eligible application. So again, everyone has its chance. Every application will be looked by independent expert against criteria and will be assessed on their own merits. So we have seen in the past that even some application with excellent names didn't go through just because the application was not good enough compared to others. So again, it's the chance for everyone. It's a competition. So without entering more, I think I will ask Oliver to give the presentation on the topic text that I'm sure that you already know well, all of you.
Thank you very much. Good afternoon, everyone. It's a great pleasure to be here today and have the opportunity to speak to you and most importantly also to discuss the call and get your view on that and your questions. Before I go into the presentation, I want to acknowledge, of course, the other industry partners who helped us with outlining this call text. What you know, this is Sanofi, Novo Nordisk, and we have as imaging company also Zeitz joining this consortium. So a few words on the project background. And maybe another note before I go into the content. What you will see here on the slides and what you will hear from me is essentially not more than an executive summary of what you have seen in our call text. As I will not provide any further details. We keep that for the discussion to get your questions and hopefully be able to provide here the answers that you are expecting. So what you will see here in this first couple of minutes presentation is just a summary of what you know to make sure that we are all on the same page. So project background. Retinal diseases are among the leading causes of blindness worldwide. There has been a tremendous advancement of therapeutic options for the treatment of the so-called anterior segment diseases like cataract or glaucoma, so that the incidence rate of blindness due to these diseases went down. Though retinal diseases still remain a challenge, despite some substantial progress that has been made in the last couple of years, particularly in the field of the neovascular age-related macular degeneration, or also often referred to as wet AMD, diabetic macular edema, and other vascular diseases. However, there are common conditions that remain that cannot be targeted by these treatment modalities that have been introduced. And this is particularly the dry form of age-related macular degeneration. This is familiar to the other type, to the wet form, which can be treated today and where the incidence rates of blindness are going down due to this treatment, though it's a more frequent form. The wet age-related macular degeneration accounts for approximately 10 to 20% of the cases, while this dry form is 80 to 90% of the cases. Also, diabetic retinopathy is a disease which currently can only be targeted with some limitations, particularly if we speak about diabetic retinopathy, which goes beyond diabetic macular edema, which is in scope of therapies that have recently been approved. And there are other conditions where also treatment options are limited, and we were looking into why is this? Where are the development hurdles? And what we identified in our consortium is that one major hurdle is a lack of suitable endpoints investigating these conditions in an early exploratory setting, but also in pivotal trials. You may wonder why this is. This type of diseases are diseases where the traditional endpoints of visual acuity do not really work. These are conditions where visual acuity is only affected relatively late, and you need to treat the patients while the visual acuity is still good. This is different to the indications like wet AMD or diabetic macular edema. These are indications where the development and the approval of therapies were built on improvements of visual acuity, and this paradigm would not work in a condition like, for example, dry AMD, where the objective is to avoid a vision loss and improve the prognosis. And we identified this as a major hurdle and thought that this would be a very good fit to the IMI2 program to look into suitable endpoints to address these diseases where we're looking at a significant unmet medical need. The question we are asking is a very complex one. And as always, if the question is complex, contributions from several parties, from several stakeholders are required. And we feel that this is something which can only be solved in a very thorough partnership between institutions that have expertise in the field. Part of these institutions are public institutions, other are private institutions, so also a very good fit to the IMI program. And as we have outlined in the call text, we believe that these 4 pillars that are summarized here on the slide, that these are the main contributors in such a public-private partnership. We have pharmaceutical companies who have the knowledge about drug discovery, drug development, as well as the regulatory and health technology assessment background. We have, of course, academia, which has expertise in methods to assess visual function and structural biomarkers which may correlate with visual impairment. And academic institutions also frequently have access to unique databases that would allow us to search for potential correlations. We, of course, also need hospitals and practicing physicians, as they have access to our target population, to patients suffering from diseases like dry AMD and diabetic retinopathy. And due to their practical experience, hospitals and the physicians there also used to have a very good understanding of the epidemiology, pathophysiology, or other evidence to predict a clinical benefit. And we should not forget about the patients, the users and the caregivers, who also play a very important role in establishing meaningful therapies, and as a precursor for this meaningful therapy, meaningful endpoints. And what you can nicely see here with this list of stakeholders, with this need for collaboration, this is going to be a very diverse consortium. This cannot be fixed by one institute at a university which is specialized in epidemiology, or by one eye clinic. This requires a collaboration across sectors between public and private institutions, and we are very much looking forward to see here proposals. Talking about the objectives for the full project, and I want to underscore full project. We are talking always about the full project. We are not looking at individual parts of potential project plans. We have always to consider that any proposal that is to be made has to cover the full project. So the objective is to evaluate novel endpoint candidates for retinal diseases, and we focus here mainly on dry age-related macular degeneration and diabetic retinopathy, as these are very widespread diseases where visual acuity on its own has significant limitations. The evaluation of these novel endpoint candidates should cover the technical, medical, and health economic appropriateness of these methods. So it should not be focused only on developing a certain device or evaluating an existing device in a patient population, or focusing on the health economic implications of a certain endpoint. It should do all of this. And it should bridge preclinical and clinical studies. I'm sure that most of you are exposed to drug development, and you know how difficult and how critical the step from the preclinical into the clinical phase is. And we are operating here exactly at this interface. We want to know what clinical endpoints match certain preclinical models. So if we have a finding in a preclinical model, which endpoint should we best employ to see whether this finding translates into human? And if it translates into human, what does this mean for patient benefit? In order to put a little bit more meat to the bones, we have provided in the call text a short list of methods that we regard to be potentially in scope. It's visual function testing beyond best corrected visual acuity. Best corrected visual acuity is established, it's validated, the latest since the large ETDRS trial in the 80s. We are looking for innovative, new function measures which are going beyond best corrected visual acuity. Electrophysiology could be a methodology which might make sense here to employ, since this measures visual function in a quote-unquote objective fashion. We all know the limitations of that, but it could be a methodology which might make sense in this context. Imaging methods to assess retinal structures, very hot topic in ophthalmology. How do certain findings on the anatomy side correlate with function? How does this translate? And I'm very happy that we convinced, and it was not very difficult to convince them, that Zeiss joined the consortium, because they also recognize that this is a need, that they are specialized in producing very fancy machines which give you nice images of the retina, but bringing this together with function, with patient relevance, is critical to everyone. Technology-wise, maybe less fancy, but not least important, patient-reported outcome tools and quality of life endpoints. Also very important to take the final step then of validating an endpoint, showing how patient-relevant this is. And I want to encourage you, don't think in the category of or. This is a question of and. We want to see everything. Yeah, I think the combination is probably the most powerful tool, and I'm sure that all of you have already creative approaches how to combine these methods. So what do we expect what this project means for the R&D process, yeah, so for the research and development process? The expectations are high, and we expect and hope to see that this project will contribute to making preclinical and clinical research more target-oriented, and personally to me also very important, more patient-oriented. We expect that the program should help us in more effectively pre-selecting promising drug candidates, and also support the attrition, so early discard ineffective treatment approaches before we bother patients in clinical studies with that. Avoiding development detours. Bottom line, a significant acceleration of the development process, and facilitating, again, very important to me, a patient-relevant development. You have seen in the call text also some thoughts about the architecture of the project. It goes without saying that this is something which needs to be customized once the industry consortium and the academic consortium finds its way together. So these are the cornerstones, what we envision here. We want to have an applicant consortium which is composed of applicants with a proven track record of strong clinical expertise in ophthalmology, a strong clinical research experience. So also on the applicant side, we want to see that the clinical and the preclinical aspect is very close together. We expect that it is necessary that this consortium has access to patients. I think this is absolutely critical. And also databases could be helpful and could be maybe an efficient way to gather data in an efficient way without initiating new studies. There should be public health experience, health economic experience, and there should be an in-depth understanding of state-of-the-art preclinical models in ophthalmology. We expect further that once the consortium has built and industry and the public applicant consortium has been merged into one consortium, that we still look beyond, and that we implement in this work stream an advisory panel which includes stakeholders that are not primary partners here in the consortium. And I'm thinking here, for example, on payers, regulatory agencies, and other relevant expert advisors which could contribute to this program. It's expected that the consortium suggests an architecture for the full proposal, and this full proposal has to be indeed a full proposal, so it has to address all objectives and key deliverables. Gaps are not acceptable. And the project should also include a plan for interactions with regulatory agencies, health technology assessment bodies with relevant milestones, and also appropriate resource allocation have to be made sure that this can happen, that we do not run out of resources for this important activities here. So what are the expectations to the applicant consortium? The applicant consortium should definitely be geared up to set up and run studies that are required to meet the call's objective. And a study could be a clinical study. I think we will not be able to complete the objectives of this call without any prospective clinical evaluations, but could also, of course, include database studies or other options to get a real-life experience. And the activities will be supported by in-kind and cash financial contributions from the companies. The FPA members, so the industry side, has committed, apart from a cash contribution, also to make contributions by provision of expert manpower. Thinking here, for example, just to throw out some examples, but that's not complete list and not limited to that. We could heavily contribute with regulatory experience. We could contribute, if this is necessary, with infrastructure for studies, study management, project management, statistics, data management, whatever belongs into that context. We can contribute with research facilities, and since we have company as a partner for imaging, we could also provide imaging and technical equipment as needed, which is not necessarily limited to that what is available on the market. So if there is a fancy idea to modify maybe an existing device or even build a new one, I think this is still all in scope. And other contributions as needed. What's in for you? I think you have probably find your individual answer for that, otherwise you wouldn't have made your way here to Brussels. Of course, there's a strong benefit from these collaborations in gaining an overview beyond the academic side on the research field and maybe even beyond that. Building interesting research networks. I think this is something which already during that phase of the project starts, that there is a discussion about who could do what together with whom. It could bring you funds for investigational studies, so I would expect that with many of you there is a lot of synergy in terms of the interest, and maybe the one or the other study is already in your plans. We want to, of course, also publish these outcomes in high-ranked journals. We're also looking for small and medium enterprises. If they want to join that here, also they could receive valuable input for their research from very experienced entities. Could also build relationships with key players in pharmaceutical industry and beyond. And they could find new opportunities for collaborations and joint ventures. I also talk very actively to a patients organization, and I think this is very important since the overarching theme of this call is patient relevance. So it's very important to stay in touch with this most important stakeholder in medicine, probably. Key deliverables will be the generation of robust data from a retrospective and or prospective studies, serving as a basis for a discussion on regulatory acceptability of endpoints for future clinical programs. Interactions with and advice from regulatory authorities needs to be thought on early on and during setup of the studies, and that, as I mentioned, would be one contribution which can very naturally come from pharmaceutical industry, since we are very experienced with dealing with regulatory authorities. The research program has to deliver data on the technical evaluation of the methods. This includes the typical parameter like validity, repeatability, reliability, interpretability, and translatability of data. We have one focus here on the translatability from preclinical to clinical development phases. And also, again, this theme of patient relevance, how acceptable is this to the patient suffering from this disease? Development of novel methods and tools is an option. For example, we could envision that a patient-reported outcome tool could be output here, or a new modality to assess visual function. The clinical validation, so the prospective study in patients with dry AMD and diabetic retinopathy. Ideally, we would like to have evaluated multiple methods, maybe even head-to-head, to see which method is the best, or is it maybe a combination. Biomarkers is in scope, particular with the objective of identifying certain high-risk populations. And whenever there are synergies between these diseases, they should be leveraged, though we have also the expectation that we are able, after this program, to come to some conclusions on both of the diseases individually. With that, I would like to thank you, and I would turn it back to Nathalie for, I think, moderating the discussion.
Thank you very much, Oliver. So I think now we can open the floor to any question you would like to ask to get clarification on the objective, the deliverable, the impact. Yes, there is a... Oh, sorry. First, I will ask you to wait for the mic here, this person. I will ask you to state your name, where you are coming from, and your questions. I think it helps everyone as well for this networking.
So my name is Ulrich Giese from Austrian Institute of Technology, and I have basically two questions. The first, I think the interpretation of endpoint you say, I think you mean more to find an early point about the progression of disease and then an endpoint where the disease has completely developed. Is that correct?
We are looking essentially at two categories of endpoints. Yeah, we have the early ones where the focus is on how do certain maybe preclinical data translate into clinical outcomes. Yeah, this is if we speak in study phases, could be referred to as proof of concept studies. Yeah, that's the one part, and then we have the late stage endpoints where we're looking in patient relevant outcomes for example, a phase 3 study.
And then I feel there is some discrepancy. What you said, at least I feel some discrepancy when you said what the main thing of the consortium should be. You said running preclinical and clinical studies. There's no word on having new technologies and biomarkers, and when you later come back to the K deliverables, then suddenly biomarkers are there and some new technologies. I think it has to be clarified what really is the sense of it.
I don't see that this is really a disconnect. I think this is a question of combining this in a creative fashion. I think from a perspective how this is to be operationalized, we speak about studies, yeah, but the outcome is not the outcome, or the objective of this call is not the study. Yeah, this is only, goes without saying, is only the method. Yeah, and within this study, we expect that these endpoint parameters are looked at. This could be biomarkers, that could be functional endpoints, and I think here it's also important then for the applicant consortium to have a proper prioritization, where to set the focus on, and what is most likely lead to fulfilling the objective.
Another question there?
Hello, my name is Lena Arfeld. I'm Regulatory Affairs from a Belgian biotech ophthalmology company. I have a question about the involvement of the regulatory authorities. So in a project, if we will evaluate on endpoint, what about, for example, the validity of primary endpoints in regulatory submissions? How would the interface be with the regulatory authorities on a project like this compared to that we as individual companies do individual scientific advices to get buy-in on primary endpoints for ophthalmology programs?
Yeah, I think there is maybe I turn this then also to Nathalie. I think there's a general response to that, because the IMI program is of course connected to regulatory agencies, namely the IMA in Europe. Specifically, we envision that we build something what I called in the call text advisory panel, which includes the regulatory agencies as advisor to give the consortium advice and guidance what would need to be done in order to show that an endpoint is acceptable, also to kind of vet the studies, if then the consortium is putting together studies, to go to the agency before the study is initiated and ask them the question, if we would do this study and provided we have a positive correlation here between quality of life questionnaire and this imaging endpoint here, would that be then sufficient for you to accept this as a primary endpoint? I think this is built in, this is this component which in call text referred to as advisory panel. And the same with HTAs, which is the same story.
Maybe I can add to what Oliver said. Coming actually from before joining IMI, I used to work at the European Medicine Agency, so I know a little bit about regulatory. Just to say that the call, first of all, there is a huge interest from the regulators as well on this topic, because indeed we are lacking the endpoint for the development of new medicines. Now, what Oliver says is that you see that in the call text there are two mentions, not only about the advisory board where we expect to have representation there, but as well a plan for a strategy for interaction, because you are probably aware that there is this qualification advice and qualification opinion. So whatever would come from the project in setting up a strategy for developing the endpoints or the combination of method, that will have to be discussed with the regulators in an advice, but as well to bring all the elements which will be used afterwards to see whether actually there is a relevance or acceptability for what has been developed by the consortium, in order to have them accepted from the regulatory standpoint. And that will certainly be as well with respect to the HTA. And you know that there is this possibility as well to have the not only the parallel advice between HTA and EMA, but as well EMA and FDA. So I would expect that the consortium will have this strategy implemented into the project on all these steps to move forward to that. There's a question in front.
Walter Luijten from K Leuven. I have a remark and a question both pertaining to pathophysiology. You said that clinicians were included in the project because of their insights in pathophysiology. Now, I highly respect clinicians. I'm an MD myself, and I think they are definitely there for a good reason, but I don't think they have some superior view from their professional, let's say, vantage point into pathophysiology. In fact, most clinicians do not excel in understanding the pathophysiology of diseases. Other groups that, like researchers, I think who make a much more sensible contribution there. My question pertains to pathophysiology and its role in this whole proposal. It seems like this is a very empiric proposal, and if one were to understand more about pathophysiology, it might be easier to look at early steps in a, say, pathophysiological cascade to see, where you can pick up the first signs of the disease, rather than looking at functional effects on vision or retinal morphology, which seem to me much further downstream from the initial pathophysiological steps in the disease process.
You're probably right with that notion. I would not categorically say that a pathophysiological project which looks into a certain signaling cascade or so, that this is a no-go area. I think that depends very much on how this is embedded into the entire proposal. But as you were saying, the focus is here on the later steps and seeing how this translates into patient relevance. To your earlier point, I would not dispute this, that if we speak about pathophysiology in a sense of investigating cascades and signaling processes and disease processes, that this is way better in the hands of a basic science-oriented researcher. The reason why this is mentioned here in the category of practicing physician or hospitals is probably because of the fact that this is coming late into the play. And if we talk about methods, these novel methods need to be tailored to the pathophysiology, but from a clinical standpoint. Yeah? And if the clinician covers that, fine. Otherwise, you remember the other pillar here is academia, which is inclusive, of course, of both, of physicians and researchers. So if the consortium feels that it is important to have a pathophysiologist in a broader sense involved in order to bring this expertise in, that's probably a very reasonable thought.
Any other questions? In front?
May I go first?
Yeah, okay.
I'm Nathalie Seigneuret from the Rotterdam IOS of Rotterdam Ophthalmic Institute and Rotterdam Eye Hospital as well. My question is, do you confine yourself to ophthalmology? Because in diabetes, I know that nowadays there are some drug companies developing drugs for systemic treatment of retinopathy. But do you strictly confine yourself, your endpoint search, to ophthalmology, or are you going more beyond to other systemic endpoints as well?
There's a call which is on retinal diseases, so it's ophthalmology. Though if there would be a biomarker, which is a systemic one, be it a genetic one or a serum biomarker, which highly correlates with an outcome of the eye disease, I think that could be in scope. But it should not be something which correlates then with kidney disease also. This needs to be tailored to retinal diseases. But as long as this systemic parameter is correlated to the eye disease and maybe mainly correlated to the eye disease, I think that would be good.
Thank you.
So there was a question in front. Here, yes. I think we.
Thank you. Mauro Placchi from Volt Pharma. Again, on the biomarkers, how do you evaluate the position of the biomarker in your endpoints? Because you know very well that there are biomarkers associated with the natural evolution of the disease and other that can be associated with the response to treatment. Now, this means that you have to do a lot of validation assessments for this consortium project.
I think this very much depends on the context, how these biomarkers are put in the context with the objective of the call to deliver predictive and patient-relevant outcomes. If this convincingly can be brought into a proper context, I would say that even extensive biomarker validation program would not be completely out of scope. The question is how likely that is, and I leave that here open, how likely it is. That's probably something where the applicant consortium need to think about. It's always important when on this call is work that don't lose sight of the overall objective. But as long as I said to the colleague before, even a pathophysiological project, which is at the first glance very far away from the patient, could make a lot of sense as long as it feeds in a meaningful fashion into work stream to deliver the objective of this program.
When I understand you correctly, you're much more interested in bridging from preclinical to clinical research. So you're going to test compounds. But you said you're not very interested in pathophysiology and pharmacology, so you're going to have not a target-based approach, but are you then interested in a phenotypical approach in animals and then bridging to the clinics? Could you have clarified that?
I'm not sure whether there's maybe misunderstanding. I think what's not the case is that we say we are interested in these bridging from preclinical, or primarily interested bridging from preclinical to clinical. That's one of the pillars, where we say we want to learn more about proof of concept endpoints. That's one pillar. The second pillar is patient-relevant endpoints for the late stages. And this may be very different. This may be different work streams that may not be the same. The reality today is that if you want to do a meaningful proof of concept study in the retinal space, you have to work with best corrected visual acuity, so with the patient-relevant endpoint. And it would be very desirable to have more reliable early endpoints where you know, okay, if I see a signal here, I will, on the back end, have a certain probability that I also see something on the patient-relevant side. So this may be two different work streams. They may lead to different results. One endpoint may match these early bridging endpoints, the other endpoint more the patient-relevant endpoints, but we want to have both in this project.
Yes.
Mahmoud Bagat from the Twin Core Center for Clinical and Experimental Infection Research in Hanover, Germany. Coming back to the point biomarkers, this is pretty complicated because there might be biomarkers that detect genetic predisposition for developing retinopathy, and there might be also biomarkers that that detects early development of the disease. I would think that these are two different groups of biomarkers. In differentiating between them is really complex. So which biomarker you would think that would be useful to your consortium? The one that describes genetic predisposition or the one that early gets the disease development?
It needs to be linked to outcomes. You need to link it to outcomes, and as I said before, you could now have a question of what we personally believe is more promising here, right. But I would not like to preempt here the creativity of the consortiums to think about what they believe is the best path forward. Though if you talk about biomarker, is as you said, is a very broad spectrum of
yes.
Of parameters
Right.
that could, even if you define it very broadly, even some of the imaging modalities in ophthalmology could fall into the category of a biomarker,
Right.
if you use a broad definition. And you need to boil it down. You can't cover this entire spectrum of biomarkers. I think that's not the expectation.
Right. Yeah.
The expectation is that biomarkers, selected biomarkers, are used as a vehicle to meet the overall objective of the call, which is outcomes.
Okay.
Yeah?
Thanks.
Any other question? This is the time for you to ask. No? Yes? Another question?
I am intrigued about your insistence on the biomarkers because talking with an agency like the EMA, well, they consider the biomarker as surrogate endpoints. So they are more propense to consider clinical outcomes. So already your project starts with this, let's say, unpleasant situation. What do you think about?
I think surrogates. If you talk now about surrogates, which I would regard as only a small subset of biomarkers, can be a candidate for surrogates. Many biomarkers, it's already today clear that they will not qualify for that. Surrogates is a challenging topic, though it's at the same time a good topic, an important topic. Several developments would not be there without a surrogate, and in ophthalmology there's one example, which is IOP, which is the intraocular pressure, which is essentially also a surrogate. Without this surrogate, there would be no glaucoma therapy, and I could envision that some of the diseases where we still have no treatment available today, they also can only be addressed if a surrogate gets accepted. And therefore, I would say, let's get it done. Yeah, let propose parameters and convince the agency that this is the only way and that they have to live with the surrogate, despite the shortcomings. They are there, yeah, and everyone knows that. But as long as we can provide sufficient evidence and sufficient certainty that that these surrogates have a certain level of reliability, I think we are good. Yeah.
If I may, actually, it's considering all this discussion and difficulty, I think that again re-emphasized the need that any consortium, the winning consortium, will have to really interact with the regulators in the sense of having the input on whether the strategy follow it seems to be sound, and whether it will generate the sufficient information and data and evidence that needs to be looked at. And again, and rightly said, is that maybe we need to think differently, and again, this is the whole opportunity to really build this within the consortium and within the project during these 5 years. Questions from, or everything is clear? If there is no question, maybe it will be more afterwards. I would suggest that we move to the elevated pitches, and then we can come back to the question, so that may still stimulate more reflection from your side. And again, take the opportunity that this is really the place now that we have the chance to have Oliver with us, to get all the clarification that you would like to have, all the debate on this interesting and important topic. So the way the elevated pitches works is that I will call one by one the person who have expressed their wish to present, as I said, in 90 seconds, and I've got the chrono here, so unfortunately I will have the bad task to remind you when we are reaching the end of the 90 seconds, to really to present briefly who you are, what you want to bring to consortium. So please excuse me if I won't pronounce well all the names, but I will try to. So the first one is Professor Stefano Genna. Yes. So, sorry, I have to ask you to face the camera, and the camera is behind you. Another complication.
Okay. No.
Well, the camera is...
why don't you come to the front
here?
here?
Come to the front. Will be easier.
Here? Okay.
We don't bite.
Okay.
Is it okay from yes? I ask my colleague. Yes, it's perfect. So we'll start the clock. And
okay, Stefano Geuna, Department of Clinical and Biological Sciences, University of Turin, Italy. We have experience in the serological investigation on retinal tissue and optic nerve. The point that we have heard before is that the jump between preclinical and clinical usually relies on quantification on histology, because we have model of disease and then we quantify how a drug protect and regenerate. The point is that it looks easy to quantify histological section, but it's not easy, because it's very complex, and quantification in histology, we know now that often is biased. And using unbiased methods, sometimes we can have the correlation between anatomy and function that sometimes is not present because of the problems in jumping between anatomy and function. So we have expertise in GLP context. We work already with companies of serological quantification on histological tissues and whatever can be measured. Thank you.
Thank you. Well done. 20 seconds left. Thank you. It makes my life easier. Thank you very much. The next one is Dr. Ulrich Gieser. Yes.
ladies and gentlemen. So my name is Oliver Zeitz. I'm representing the Austrian Institute of Technology. It's Austria's largest non-profit organization, and it mainly is a technology and innovation provider doing mainly applied research. And the business unit Molecular Diagnostics I'm working for is divided in 5 working groups. That's biomarker development using next generation sequencing, epigenetics, and autoantibodies, bioinformatics department doing all the analytical tool for that, and the assay development and validation working group I'm leading for, which transfers the results in reliable routine assays. And then we have also a sensor development group, which is focusing on optical sensors and electrochemical sensors. And they are currently developing an optical coherent tomography device, which is much smaller and much cheaper than the currently existing ones. It's movable and can be used for mass screening devices and for mass screening of people. And then we have also a system integration group, which combines all that into one point of care device. And we are looking to joining a consortium to bring out our innovation and technology experience. Thank you.
Great, thank you.
Yeah.
Thank you very much. The next speaker or the next person will be Dr. Muriel Boucard.
I prepared a PowerPoint, but I was told that we cannot use it. So I am Muriel Boucard. I work in Lille University Hospital. So I have a group of vision scientists and clinicians. We work on various pathologies like AMD, glaucoma, Stargardt disease, and normal aging, visual problems in normal aging. Our equipment is based on reaching and grasping tasks. We have also a panoramic screen, 5 meter diameter, and so we investigate object search in panoramic dynamic viewing conditions, eye movements in natural actions, reaching and grasping objects in cluttered environment, saccadic response task, posture and balance. So we have different types of eye trackers. We have a Polemus system, we have the panoramic screen, and various equipment to investigate vision in patients. So we do cognitive sciences. Thank you.
Thank you. Thank you. I think I have to congratulate all of you to be really within this 90 seconds. That's great. The next would be Professor Stefan Pohlmann.
Good afternoon. My name is Stefan Pohlmann. I'm professor of experimental psychology at the University of Magdeburg in Germany. And my research focus is on visual search and selective attention. In recent years, we have investigated patients with macular degeneration. And what we have observed is that they show a deficit already early on in the disease of what we call contextual cueing. Contextual cueing means that we all typically take advantage of regularities, of patterns in the environment whenever we come to an environment that we have seen before or that is similar. And these patterns, they automatically, implicitly guide where we look. And this breaks down in AMD, and because of its obvious importance for everyday search behavior, we think that this could be a good contribution to the diagnostic endpoint that is searched in this program. In addition to these behavioral paradigms that are quite cost effective and could be used also in large-scale patient testing, we also have a very good neuroimaging environment in Magdeburg, particularly fMRI, and we also use eye tracking, which both could be used for more in-depth investigations of selected patient groups. Thank you for your attention.
Thank you. next person is Professor Ursula Schmidt-Erfurth. Sorry for my pronunciation.
I'm accustomed to this. Usually people ask me for my address in Erfurt and they can't find it. So, but the name is just one name, Schmidt-Erfurth, no village and no city involved. I'm actually from Austria. I'm the chairman of the Department of Ophthalmology at the Medical University of Vienna. And we also founded the Vienna Reading Center, which is a fully digital electronic analysis platform for looking on retinal biomarkers. So our engagement is very intensive. I would like to congratulate Bayer and particularly my old colleague, Dr. Zeitz, that they initiated this activity because we all know that vision loss is a very severe burden, not only for individuals, but also for the entire society. We also know that efficient treatment is badly wanted, but we have realized that by just treating neovascular AMD and DME, which is a small proportion of the entire field of retinal diseases, has ended in the biggest burden of treatment and the biggest budget drain in medicine that we have seen during the last year. So we have to come up with something which is efficient yet personalized and which is affordable, and that has to go hand in hand with diagnostics. Since there is no good animal model, and since man is not a mouse and an old person and a diabetic person is not a diabetic and an old mouse, we have to go to humans. We have to be non-invasive because this is repeated screening and we have to treat early. So your comment about the end stage and other comments going to the same direction is very relevant. So we have to interfere early.
Thank you. Now we'll call Professor Antonio Silimati.
As Nathalie told you, I'm Antonio Silimati. I'm from the Department of Pharmacy and Pharmaceutical Sciences of the University of Bari. The expertise in Italy, the expertise of my department spread from the computational chemistry, lead identification, synthetic strategy development, and preclinical studies. In the last 10 years, we studied the beta-adrenergic receptor, beta-3 subtypes, and its gene polymorphism is related to diabetic retinopathy. In the last years, we published and patented some compounds that can affect this receptor. So we would like we are looking to a consortium to provide these compounds for further development.
Thank you very much. Next person, Dr. Rocher Smith. Okay, I think it's not here. So we move to Professor Gracia Soveral.
Good morning. My name is Graça Soveral. I come from Lisbon, the University of Lisbon, Faculty of Pharmacy. I am a professor in biochemistry, biophysics, transport, membranes, and I do research at the Research Institute for Medicines. So my scientific interest is based on biomarkers, new targets, and as a biochemist and doing membrane transport, I think we have new targets that could be validated for these retinal diseases that are membrane channels important for the transport of fluid and fluid balance between tissues. Regarding these specific membrane channels, my particular interest is in aquaporins. They transport water and they have been recognized as important targets in several diseases because they have been found to be abnormally expressed or its function is damaged. So I would be interested in going into a consortium in order to validate these biomarkers and contribute to retinal diseases. Thank you.
Thank you very much. Then Dr. Andrew Spink.
Good afternoon. My name is Andrew Spink and I work for Naldus Information Technology. Naldus is an SME which is got 130 employees and based in the Netherlands but active worldwide. And what we do is we make innovative solutions for behavioral research, especially software. To give you an idea of the sort of things we do, I want to mention very quickly 2 products. One is EtherVision, which is a video tracking system which we think is the market leader and used in many behavioral tests for experimental animals. Another is the Erasmus ladder, which we have recently launched, and that is a specific test for testing behavioral flexibility and cerebellum function in mice. In this call, we would very much like to help you automate any behavioral tests that you want to develop for this. You can think perhaps of the visual acuity test or variants on it, but also, for instance, combining that with physiological data, which we have some expertise in. So if you're forming a consortium where you, as part of the work, need to develop new tests, either in animals or in humans, we also have equipment for measuring behavior in humans, then I'd be very happy to hear from you. Thank you.
Thank you very much. Next person, Dr. Eugenia Tagliavaca. Please accept my apologies.
Hi, my name is Luigina Tagliavaca. I'm coming from Italy. I'm part of a member participant of the Italian consortium. His name is Consortium for Biological Pharmacological Evaluation. It's a consortium established in the year 2000 in Italy and now has several offices in Italy and the Mediterranean area. The goal of the consortium is to facilitate innovation in the pharmacological and biological fields. To the aim, we provide project management, international projects, introducing new and innovative clinical trials design, and provide regulatory advice and applications. So the consortium has expertise in several areas, which are clinical research, clinical trial design and implementations, regulatory affairs, health technology assessment and pharmacologic evaluation, pharmacovigilance, regulatory application to the IMA, we focus mainly on pediatric drugs and on rare diseases, registry and databases, and also educational training. So if you need some, if I can provide any help for you, you need to contact the website. It's www.cvbf.it. Thank you.
Thank you. Last person, Ms. Stephanie Ubridge.
Thank you. Good afternoon, everyone. My name is Stephanie Ubridge. I work as a Research and Development Project Support Officer at University of Alcalá in Madrid, Spain. And I'd like to present the Biomedical Engineering Research Group, which develops basic and applied research activities in biomedical signal processing, medical devices, and sensors. They have gained broad experience in the analysis of multifocal visual evoked potentials and multifocal electroretinography, applied to the early diagnosis of optical neuropathies such as glaucoma, optical neuritis, and diabetic retinopathy. And these techniques allow for the assessment of a large cross-sectional area of the optic nerve and therefore accurate functional evaluation of the visual pathway by objective and non-invasive means. So far, several studies have confirmed this, and this research group is actually looking to join a consortium, and what they offer is their complementary vision but also their experience in international projects, the holding of 8 national patents, but also the possibility to apply these techniques to patients with optical neuropathies and test these results from clinical trials, but also compare multifocal visual evoked potentials with other methods such as MRI, OCT, or SAP. Thank you.
Thank you very much. Just want to be sure. So, Dr. Roger Smith. Okay, so it's not here. So we have finalized our elevated pitches. So I hope you find it useful. Ah, okay. A question or? Okay, yeah. Yeah, I think we have time if you...
Yes, because we submitted a pitch, but it is maybe there's
okay.
went something wrong and...
Okay, sorry, we don't have that, but please, we
yeah,
have
no
time.
Okay.
90 seconds.
Yes. Yes, I would like to present Rotterdam in the Netherlands and the Rotterdam Ophthalmic Institute. Today, my name is Nathalie Dorstein, and together with my colleague Koen Vermeer, I represent the Rotterdam Ophthalmic Institute. This is a non-academic research institute in ophthalmology, and we are part of the Rotterdam Eye Hospital, which is the largest and the only one eye hospital in the Netherlands. So in our collaboration with the hospital, we have a huge patient population. And for that reason, we are very experienced in clinical research and have good experience in contract research phase 1 until 4. But that's not all. Moreover, we have a research group in imaging and image analysis. And that makes us a very special group because we have the combination of the patients and the technology. And for that reason, when I go through the list on page 16, I see that we belong to the others of the others, maybe the last bullet. So a non-academic research institute with good experience in clinical research, but on the other hand, the technical experience of imaging and image analysis colleagues. So we are looking for the other partners who can share our experience in this consortium. So thank you very much.
Thank you.
Thank you very much for the opportunity.
I think we have another request from Victoria Dominguez del Angels.
Thank you. My name is Victoria Dominguez del Angel. I'm the CEO of Abegenomica company. It's a small company from France. We are a company that it's jeune entreprise innovante and then make 60% of research and 40% of service. And we are involved in data management content for the big data. And it's our specialty, and I want to invite to contact us if you have a management of big data, well, all these consortium that create. And we are specialists in 4 points. The first one is about data center. We created or we can use the data center in the cloud computer form. And also we use all this security data that you need it if you want to be sure that your data it's in a good use it in a good way. And the last point that for us is very important, the reproducibility of all this information, that you can use it, for example, if you want to share with your partners, and also in the case that in the publish your pipeline or all these things that can be useful for the others. And well, I think it's everything. And, please, you can see on our website, avegenomica.com.
Thank you.
Thank you.
Thank you very much. Now, is there any, we still have a bit of time, so is there any one who wants spontaneously do this yeah, okay.
Not spontaneously, I applied, and I even got some correspondence with you.
Oh.
Anyway, so my name is Alexander Dömling. I'm heading the Department of Drug Design at the University of Groningen, which is in the north of Netherlands. And I'm a chemist by training, so let's see. Let me prepare the floor. Personalized medicine was coined some years ago to account for the increasing diagnostic coverage of diseases of each individual and pave the way to individual treatment. While diagnosis is soon becoming daily reality with next generation sequencing, the treatment options are not at all elaborated. Hereditary and spontaneous mutations are responsible, however, for a great majority of diseases. However, almost no treatment options are available for mutation-caused diseases. I'm able to reverse major deleterious mismutations on the protein level by a pharmacological approach using small molecules. And this is not a chaperone approach. I target mutations, for example, in age-related macular disease, diabetic retinopathy, cataract, just to name a few. I believe that my approach will be a game changer in many areas of medicine, especially with exome sequencing, and has the potential to truly fulfill the promises of personalized medicine. I'm looking for clinicians in ophthalmology who can help me to select the right diseases. I'm looking for experimental ophthalmologists with the right cell and in vivo models. And I'm looking, of course, for companies who help me to develop our compounds. And at the same time to develop the corresponding diagnostic tools. Thank you very much.
Thank you very much. So now I look at my colleague because I don't know if I have the updated list. So do I miss anyone else? Ah, there is one.
Good afternoon. My name is Sergio Larreina. I work for PONS, which is an intellectual property agency located in Madrid, in Spain. I am in charge of the Competitive Intelligence and Technology Transfer Department. PONS has more than 60 years of experience advising on and defending IP rights from our offices in Spain, San Francisco, Bogota, and here in Brussels. We are specialized in the field of industrial and intellectual property rights and new technologies law, offering professional advice in designing and implementing strategies to create, maintain, defend intangible asset portfolios like patents, trademarks, designs, copyright, domain names, etc. in every country. Our services also include economic valuation and consultancy reports, analyzing technology trends, markets, key players, and possible scenario in order to provide a complete picture for a possible R&D investment. We offer our experience for any consortia looking for legal, market, technology, and IP right advice. Thank you very much.
Thank you. So have I missed someone? No. Is there any spontaneous person who would like to as well do this exercise? No. Is there any more questions? Do you have any more things you would like? We don't seem to have any question coming from the web. Any more question? Yes, please. We still have time, so please feel free to really use as much as possible this opportunity to
really one question, Dr. Zeitz. Could you summarize your final goal of this project in one sentence, please?
In 90 seconds, or?
It's in return now. So the key objective is to identify predictive and patient-relevant endpoints. you can criticize it as being generic, but I think that's a very important, medically highly important topic, and we need to fill this with life.
Now I have a question, because the topic addresses both the dry AMD and the diabetic retinopathy. How much do you expect from the proposal to have like 50% on one, 50% on the other one? Or is that something you have thought about? What would you put the weight on, or on really the two issues to be really considered?
If you would force me to give you a number, I would probably come up with a 50-50 split. However, in the context of this call, I think you can view these two indications also as type of model indication. Yeah, model indication for diseases where no treatment is available and where visual acuity as an endpoint does not work. Yeah? And there are potentially other diseases where you could extend that. And if a proposal would bring it to a point that this endpoint topic is worked up so generically that it can be generalized across these two diseases and maybe even beyond, that would be fine. Though I would expect that this is probably not going to happen, and therefore we need to focus on these two diseases, and you should try to find a balance that is kind of equally weighted.
Okay. There was one question here.
Well, just a comment on the one single great objective that you made. Should this be applicable on Western countries, or should these measures be also applicable in poor settings where you don't have sophisticated technologies to monitor or predict disease?
I think the ideal scenario is that this can be generalized. Yeah? That is not something which works only in a specific environment, in a specific ethnicity or so.
So it has to
be. It depends. Yeah, it depends on what the actual idea is. I would say if you have something which works well in Caucasians but not in Asians, for example, that's not something which I would throw right away. But ideally, it should be generalized.
Okay.
So it's here, yes. The mic here, please. The mic.
I think that if you repeat the context of your project, probably everything will be clearer. For instance, first of all, stress the objective to reach the endpoint. Because when you have explained before that the objective, it is the endpoints, well, for me, this was the answer partial to the second question that I raised at the beginning. So the objective, first of all, should be defined clearly, and then from the objective, going to the endpoints where you stress the measurements and the timing at which you want to achieve that.
No, I think the objective for the purpose of the call is relatively clear. I think what now the next step is to fill this call with life. We left several points very intentionally open in order not to limit ourselves too much right from the beginning. Of course, it would maybe be an easier task also for the consortium to find together if that would be more set up in a checklist way, where we say this biomarker, this imaging endpoint, this function endpoint, please do a correlation study in 1,500 AMD patients and in 1,500 DR patients. That would be easier. At the same time, I would think it kills creativity. And we essentially do very much hope that during this process, we are getting exposed also to new ideas that maybe the group which is currently working on the call has not on the radar. And we would very much hope that we see, and I heard a couple of really new ideas and new thoughts during the short presentations during the last half hour, which no one in our consortium had on the radar, and we want to hear that, and we want to pull that together, and therefore you can criticize that, but for that reason the call is in some aspects open-ended and maybe a little bit vague and unspecific, but the intent is to leave you, and in the end all of us who will run this project, a little bit of freedom and to be open to new things.
Yes, but freedom should be associated with rigor, otherwise the data.
Last opportunity for a question. If not, I think indeed I want to emphasize the fact that this call, if you have been familiar with other topic on the IMI1, there was much more some suggested architecture, much more describing already proposed work packages on how to work. This is much more left open, and indeed this is really to bring all the ideas that maybe since it's a moving field, we don't really know, it's to bring and to really maximize from all the ideas, and this will be reviewed by the evaluators when during the process of the evaluation, to really find out what could be really the best application to move forward and to work with the industry consortium. So since we don't have any further question, I would like to thank Oliver, to thank all of you. Yes, Matthias?
Maybe just, I think it will work without the microphone. Just one additional information that may be of interest for you.
The web. For the web stream, sorry, I think it helps a lot.
Matthias Gottwald, Bayer Pharma. Maybe just one information that might be of interest for you is you have seen the participating companies on the intro slide from Oliver. There is a very high probability that at least one, maybe two other big pharma companies will join this project during the next weeks. So don't be surprised if there will be even more resources available from the industry side in this project.
I think, thank you, Matthias, for this clarification. I think it's great. It shows the interest around this topic. Now, in terms of, for you to know, is that any additional contribution will come from the industry side, but from the public side, the contribution is set in the core text. So that, of course, will help with further work, that's for sure. Now, I would like to say that since there is still a bit of time before the deadline for the submission, which is the 12th of November, in case you have any question, please do not hesitate to contact me. I'm the scientific officer in charge of this topic, and therefore I will follow from, I'm following this topic from now on until the end of the project, and therefore, please, this is, if you have any question, come directly to the IMI office to send the questions, not to Oliver. I want to insist on that. Again, it has to be clear. There's no communication between the potential applicant and the industry consortium during this phase. This is really all channeled through IMI, so please send me your questions. I've been cut, no. Send me your question if you have any things you would like to discuss, both on the process point of view as well as on the, this particular topic. And on this note, I would like to thank you very much. Just to know, for you to know, we'll have the coffee break, so since we have finished earlier, the next session will start as well earlier than expected, so instead 4:30, it will start now at 4 o'clock. You still have more than half an hour for the coffee, so plenty of time for you to network, and please use this opportunity to really, now that you have heard a lot from everyone, to network during this coffee break. Thank you very much.